everyone. Thank you for joining for this session. I am Nakshatri. I'm from Indiana University School of Medicine. I basically work on breast cancer.
Hi, everyone. I'm Mariana Boroni. I'm in the Brazilian National Cancer Institute, leading the bioinformatics group. I mainly work with gynecological cancer.
Okay, so we have prepared few slides. It doesn't mean that we want to take away all of your time for the discussion. Just to set the stage for the discussion. Again, this session is recorded. So being here, you consent to be participating in this process. So we have a couple of objectives for this session: disparity in incidence and outcome. Many of you know, or various diseases, including cancer, is well known. We always focus on the socio-economic factors, but the emerging data suggests genetic diversity shaped by the evolutionary history, environmental exposures, and demographic shifts contribute to disease. And can we use the latest technology to address this possibility? So why, hopefully you can feed us back on saying why this explore better way to incorporate human diversity in cancer studies. I will keep this open later on when we come back to discussion. So the question is, why do we have to?
They were saying that they are seeing the notes in the.
Okay, what do I have to do?
So maybe, I don't know, maybe
This one, that's the way it should work.
No.
Okay.
Display settings, maybe?
Yeah.
No.
Okay. So duplicate the search or no, right?
I'm not sure.
I don't know what the problem is, but
Do you know how to fix? No. Duplicate slide.
Okay, that should do it. Okay, now it looks okay. Good. Okay, thank you. So many of you know the Out of Africa Foundation for Genetic Diversity, Human Origin in Africa. Then we have the genetic diversity. Then we also have the adaptation to various environment, and that is giving us to the modern disease relevance. And some of you may have seen this paper which came out in Science many years ago, where people have identified the different loci that is responsible for the adaptive traits, which influences our response to marine diet, lactose tolerance, malaria resistance, cholera resistance, arsenic-rich environment, cold climate, altitude, light skin, and short statures. And some of the genes, they have the GWAS in these genes have been linked to various phenotypes that we have. Because of these adaptive traits, in addition to our genetic variants, which makes us who we are, so humans are very different. So that's why we need to critically include diverse people in genomic studies to ensure that all people benefit from this knowledge. So this is something we need to take into consideration, particularly in the context of cancer, evolution, demographic history, and environment shape human variation. Again, this is a very interesting paper which came out last year in Nature Medicine, emphasizing why we need to use multiple samples from different groups. Genetic ancestry also influences disease risk, development, and drug responses. That's why the theme of this meeting already, I don't have to say again, underrepresentation of diverse ancestries limits our precision medicine. So biospecimen collection, we should focus on diverse population. And the one thing which is really has gained a lot of attention recently, this is an article, it came out in New England Journal of Medicine in one of the traits called Duffy, which is more common in African ancestry. It is in the population because it protects against malarial infection. But what has been emphasized in this particular article in New England Journal of Medicine is these individuals naturally have lower neutrophil count. If you have a lower neutrophil count, you are automatically excluded from clinical trials. And the drugs that are given, chemotherapy is given to them is also lower. So that really affects a section of population which are getting the cancer treatments, right? That's why there is a call for more research into how this germline variant, this is one of the examples. There are many other examples, variants affect cancer in general, and in this case, breast cancer biology. So with this background, we are going to go back. It's not going back. Okay. I'm going to put up the main questions we had before. These are the questions we have in our mind. Again, any other thought process you have, I'm not going to read again. I will keep it as it is. And it's open for discussion now. Please go ahead and have your thoughts on these questions. How do we address this?
Yeah, as you may heard, Human Cell Atlas is moving forward to include also Disease Atlas. And our main goal here is to think about how would be this cancer-related atlas. So how we envision this atlas to be a representation of diverse people, diverse cultures, diversity of exposures. How do you guys think about it when we think about a cancer-related single-cell atlas?
Should we go question by question, or you guys want to take up any one questions and some of you want to elaborate on that?
Yeah, maybe we can go question by question. For instance, what would define a comprehensive cancer atlas combining single-cell and spatial data? Should we think about matched tumor and normal cells? Thoughts on that? Only tumor cells, or should we require to have also matched normal cells?
I think we should have normal cells too as a reference, because anyway, when you do experiments or science, you need to get something to control or something that you can reference. And then in the future, if these data sets are going to be useful for other people to compare to, they have something that they can draw out meaningful conclusions.
But when we are talking about normal cells for comparison to tumors, what would be the best control?
So say, for example, if you do colon cancer screening, like in the States, you would have a long-term follow-up of that patient, like in more developed country. So those are, so say 60 years old and above, you started to screen the patient for colon cancer, and the biopsy results can come out to be non-tumor. So those can be used as healthy controls, and we can save those data because these are available in the, in the medical center somehow.
Well, just to make us think about it, do you think the cells that we have already profiled in the HCA, they could be used as these normal content parts?
ETC?
In the Human Cell Atlas project in general?
Yeah, for example, the normal breast atlas has been already generated by HCA with the different groups. Should that be a normal reference, and making sure that this normal atlas includes the samples from the diverse population so that we have a uniformed atlas that can be used as a reference for all the cancer atlases?
I think so, but then it has to be specified what the race, age.
Oh yeah, I totally agree. I think there are case to case where it's very hard to also have the normal content part, right? So for those, then a separated cohort would be the case control, but when possible, having matched samples would be very good. And what about representation of the tumors? What we envision as a good representation would be one sample collection, would be several sample collections in terms of solid tumors, for instance. What do you think?
And there is also an additional layer of complexity because nowadays the neoadjuvant treatment is becoming a more common form of a treatment. So the pre-treatment materials are becoming incredibly difficult to get as well. So that is something we need to take into consideration as well. Sophia, you have an answer to these questions.
Well, just a comments. It really depends on the It really depends on the tumor type, on the disease, and accessibility, standard of care. So of course, pre-treatment, post-treatment, during treatment would be would be great, but if the standard of care is not to have repeated biopsies of the solid tumors, then you're only going to get what you get at the time. And so I guess it's important to work closely with your clinical colleagues to be able to capture those tissues. And similarly for the pre-cancer, yeah. So for the pre-cancer lesions, so my point, my idea and thought is bank every possible stage and state. But for some diseases, you don't get the pre-malignant lesion, within intentionally. Sometimes it's by chance because it was a different surgery for some other indication and pre-malignant lesion was identified.
So, considering the points raised, should the spatial be the priority where you can do it with the pre-treatment biopsies, whereas the single cell studies may not be feasible, hoping that the spatial will become cheaper and easily affordable for everyone to do everywhere. You have some experience in working in the pre-cancer lesions, do you? Yeah, probably you may be able to give us some insight.
I think for head and neck oral lesions, you can get pre-malignant lesions, you can get matched stage 1 through to 4, but it depends. I think, we use hospital grade FFPE, so you have to use what they bank. And
Yeah.
I guess, when you look at the Cancer Grand Challenges call this year, it's on TME dynamics is one of the main themes. So they really want to know not just single time point, but over the course of therapy. But to your point, with neoadjuvant IO coming on board, we're going to get less tissue up front. We're going to get on treatment tissue. So we have to bank and store, and also, tissue at autopsy. So, maybe that's a space in which we should start to biobank, I guess, or access those cohorts because you have that time point, and then you work backwards to what you can. But I think increasingly we'll get on treatment.
Yeah, you raised another important point. How many institutions have the heart autopsy programs for cancer? That is another way of looking at where we can build the whole thing too. How many of you have it? We have it for the lung cancer. Okay. So there are few institutions have this, but it's not universal. It's pretty expensive to have that program. That's another place one can think about having a comprehensive atlas done. Okay.
In terms of technology, I think we talked a bit, but maybe we can explore more. What would we, as a community, prioritize? Spatial, single cell, single nuclei, panel, if panel, what would be the gene markers? What do you think?
One thing we all need is a cheap technology which can do everything, right?
Yeah, let's imagine that budget is not our problem.
So anybody who thinks that the spatial is the way to go otherwise, or going back to the single cell nucleus, which one do you think you prefer in this space based on what we discussed?
I'm going to give my personal view on that.
Okay.
For instance, I work in the Brazilian National Cancer Institute, and there we have a huge biobank of FFPE samples. So thinking about joining metadata and generating data, this biobank would be the best case scenario because I know already what happened with the patients in terms of clinical outcome. So, and thinking on the type of sample, spatial transcriptomics would suit the best. So ideally, I would go, for instance, for spatial transcriptomics.
Maybe you can follow it up with the single nucleus RNA-seq if you have enough resources. Maybe the spatial comes first, then the single nucleus, because most of us have either frozen or FFPE samples in which you can't do the single cell, maybe single nucleus, maybe the next one to consider maybe.
But do you think this would be the same scenario for most of you, or you
Or
guys?
any other technique do you guys are aware of which may be applied as well? I know the AI-based characterization of H&E slides are becoming a major popular move now. So maybe the third category, or integrating these data with the AI-based H&E staining slide, maybe make it much more practical to everybody. Yeah.
I mean, if one follows the field to date, it isn't in spatial transcriptomics, right? And if one follows clinical diagnostics, it isn't spatial transcriptomics. So I think one has to evaluate what you want the comprehensive cancer atlas to be. If you just want it to be descriptive, then you can go after single cell or spatial transcriptomics. If you need it to be clinically relevant to current therapies and diagnostics, it has to be padded on H&E and proteomics first, right? These are not cheap assays.
Yeah.
And the ground truth, I think this is what we never had to deal with single cell RNA-seq, is the ground truth has already been established, which is protein and H&E. So removing yourself all the way downstream to spatial transcriptomics, it's just, as an atlas, do we just want to provide all of the things? If we want to provide all of the things and allow clinical to come, further downstream, that's okay. But that has to be kind of clear from the top layer, right? Because those are two different worlds. And the world for comprehensive cancer atlasing is already being done in spatial from a very large initiative. Kind of most people, including people like Arutha or myself or Sofia, are already in the proteomics land, right? So if we are kind of playing catch up, or we're just going to say we're not playing catch up, we want to be purely discovery, we want it to be foundational, it can apply to clinical later. And I think until we really think about that and focus on that, all the other points are mute, because everyone's going to have their favorite platform.
I mean, in my opinion, we need to focus on the foundational side as well, so that we can distill it down subsequently to a clinically applicable markers, right? So where you bring it down from the spatial, you identify the markers which tells something about the disease phenotype, which somebody else will convert into a H&E and IHC based staining subsequently downstream. I mean, in my personal feeling, neither the spatial or single cell is going to be a day-to-day clinical diagnostic tool. It's, unless they make it for $1, you can get everything done in an hour or so, which is not going to happen.
Yeah, but that's generalizables. Like,
Yeah.
what is spatial? Spatial can be one IF stain, and that isn't a
Exactly.
billing code, everybody.
Yeah,
Like,
exactly.
it exists, right? So H&E is spatial.
Yeah.
And so I think we have to be very clear about our terminologies and goals, right? If spatial omics is just descriptive and all of the things and gene expression, then that's okay, right? But it is clinical already, right?
Yeah.
It single cell never was. So that's what I'm saying is, like, do you need to match it? And saying that's not there yet, it already is, right? So.
Based on your comment, what do you think would be the minimal data that a comprehensive atlas should have to address this point?
I mean, everybody needs to do an H&E no matter what you do downstream, right? I think that's top tier. That's kind of like quality control. I mean, I don't know, you can't generalize a panel. So if you want to go spatial transcriptomics, how do you generalize that? My panel is going to look different than Arutha's because every cancer is different. So, I mean, I think HCA is so heavily based in being able to go to Ari's point of foundational models. We have so much single cell data. We need to leverage that understanding, and then maybe that spatial transcriptomics.
But then total RNA is your opinion?
I mean, total RNA has been done on tumors for almost 2 decades, right? There are only few which have gone into clinic on a routine in the precision, right? So, yeah.
Great conversations. I'm just echoing what Jasmine was saying. I think we need to go spatial transcriptomics because you have that, their whole transcriptome assays available today, but you need to layer in the protein, you need to layer in the H&E because your endpoint is not a UMAP anymore. Your endpoint is tissue in space, right?
Yeah.
And the other bit is, yeah, so it's just how we think about the other bit is on the clinical end. So clinical is very different from the discovery space. If we're going clinical, it has to be H&E based. I mean, the Acrivon Therapeutics 10-plex, which is now a 5-plex, it got FDA approval a couple months ago. It's a spatial signature, but it's just positive, 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 positive in the tumor. So they call it spatial, but it's not a spatial signature. It's IHC positive for 5 markers in the tumor, which has FDA breakthrough designation for a companion diagnostic for an ovarian cancer drug for a phase 2 drug from Acrivon Therapeutics. So there's a roadmap for spatial signatures to the clinic. My question is it clinical that is the focus or is it annotation of, yeah, so it's where do you land on that, which is a challenge, I guess.
Yeah.
This
I
is a
mean,
great point.
we can limit our discussion rather than going all the way far into clinic to the discovery phase itself at this stage. Then maybe the next reiteration will be the clinical, right? From the discovery point of view, where we should focus ourselves. Maybe that's where we have to frame our discussion.
Yeah, and we also need to think that there are some tumors that have been heavily studied, such as ovarian cancer, breast cancer, but there are other tumor types that are very, how do I say, high incidence in people who had been outside from sequencing projects, as for instance. So for those tumor types, maybe we are still in the discovery phase, right?
Yeah. Please, there's.
Thank you for the discussion. I find this really interesting because for TissueMap, which is the program in Singapore for doing spatial omics in cancer, selecting tumor regions has been very difficult. And one thing that we've been thinking about is, can you use the AI to select these regions? Because if the AI is good at predicting therapy response as reported in these papers, then can we use them to ask which regions are worth assaying? I think the flip side of that, in our discussions is that, well, then, if there's some information that's not present in the H&E, we will never find it. So I don't know what people think about this. I'm curious to know. That's the first part. The second part is here in Singapore at the NCCS, they collect something like 20 to 100 blocks of tissue per patient at point of resection for colorectal cancer, including things like the lymph nodes, as well as any form of like LVI, any invasion of the lymphovascular system and so on. I don't know, have you guys thought about that? Is that important to cover in a cancer atlas?
I think you brought us to the third question where you raised all these issues. Any pathologist in the room who can chime in and see whether we are off track or on track? Any pathologists? No, nobody is here. No pathologist.
Well, generally, when you guys are designing an experiment and then you go to talk to the pathologists, how many areas the pathologists select for you to sequence?
I mean, that has been one of the major criticism of precision medicine, right? You send the things for sequencing after what is left for you to do it rather than representing the entire tumor, different blocks. That's the reason why a lot of precision medicine-based drugs work only for a short time because you're not representing the entire tumor. So, I mean, we will have the same limitation here as well because we will only get the leftover tumor tissue for all of our spatial work or single cell, whatever the work. Please go ahead.
Thank you. Now talking about pathologists. So probably we or you might need to organize a specific meeting for them.
Yeah, I know.
Because I don't want to offend anyone if somebody here is a pathologist, but it's problematic. The communication with them is very problematic. So we have been doing multiplex imaging in tumor sections, and they don't annotate fully. We understand that they have a lot of work and burden and everything, and probably research is not their priority, but we have had this struggle.
Yeah.
And I do think it's before, so if we're going to be using sections, I do think that we have to start with them and like really tell them that they are critical for us to continue with whatever technology or whatever TMAs we end up doing. They are critical, and probably they might need a specialized section in this meeting dedicated to them for them to realize about how critical is their work.
I agree with you there. I mean, the hospital system everywhere doesn't incentivize research. So I know that's why they're in pressure all the time. So it's hard to catch hold of a pathologist. I completely agree with you. We deal it all the time. So we are into number 3 because we want to make sure that we can cover as many as possible. So, I mean, one of the things, many of us, at least in the solid tumor world, we know that the metastasis is the major killer. If we are given a choice, what should be our efforts in collecting and analyzing the metastatic tissue over primary tumors? Yeah, Sophie. Okay.
To be doing both, and again, it's based on availability, but it gives us an idea of the history of the tumors, identifying clones, and then which metastatic nodule, right? So are you, at least for the diseases that affect the peritoneal cavity, is it like you go all the way to the lung? Like, you know, how far up do you go? Which bone, brain, right? So it really based on the question. And I would say just do as much as you, what is the limitation is money.
Yeah.
If the tissue is available, then the limitation is funds.
Yeah.
And one thing I was thinking about while you all were talking, although it's not tissue, but circulating tumor cells, circulating tumor DNA. I know this is not at the tissue level or the cell level. However, though, clinically, this is what because this is the cancer section, this is what is being implemented now everywhere.
Yeah, ctDNA is the one which is relatively easy to do rather than the circulating tumor cells. That's why a lot of places are going in that direction.
And it's an FDA-approved test.
Yeah, exactly.
Body is like ordering that now for their patients, whether or not it means anything right now.
Yeah, it's a good point.
Yeah, yeah. So let's move on to the question number 4 we have. How can we ensure each sample is annotated with the rich clinical metadata? Stage, treatment response, and outcome. I know this is, it is related to how much information you can get as a researcher from the clinical data, right? So one has to have a very good relationship with the tissue bank who collects the samples for you. That aspect, at least in our case, we don't have any problem. We can easily get all the information on the tumor samples we get for our studies. I don't know whether anybody else have issues with those kind of getting those information.
Should we think about a minimum clinical data associated with these samples? So in the same way that we thought about diversity and minimal metadata associated with data, should we think about, a minimal clinical data?
I mean, in that context, I think what we may need to ask is to get the genomic DNA from that patient, the blood, which is not always collected. If you want to get to the diversity to make sure that we have the diverse population, we can link it back to genetic ancestry. That is something a lot of the tissue banks are missing. So probably if you are taking something on a research subject, having the genomic DNA from the same donor is something we need to emphasize on. I mean, that's my personal view. I don't know whether others agree with that.
Good. I was also thinking that exposition, some data associated with exposition, because when we are talking about diversity, it's not only genetic diversity, but also diversity in terms of exposition, right? So this would be also a nice metadata to come along.
Yeah, exactly. Yeah, please.
Just a comment on the multiplex IF. I think often we forget that pathologists are trained in two colors, right? And we take our multiplex data and we false color it brown to, and then we give that data to the pathologist and we say, here's your brown stain. We've got the layered 950 plex, whatever. And then they score that. So they're used to looking at brown stains all day. They're not used to looking at colored images. When you give them a colored image, they don't know what to do. So they look at H&Es or brown dab stain. That's it. So we need to either false stain, I've presented at pathology visions to 1,000 pathologists and they don't get spatial. They understand H&E, they understand dab staining. And I think we need to meet them where they're comfortable. Otherwise, the adoption curve is too steep for them. If we give them RNA data, they don't know what to do with it. I think protein's the intermediate and maybe false. And the false coloring is sort of funny, but I think we need, that's where the point of adoption is, right? And then you show them, oh, look, it's yellow or whatever it is. So I think we just need to be creative in how we can't just bring pathologists and say, look at our colored data, Yeah. No. Even with multiplex, we false color dab, and then we give them those images, and then they can score and all of that. So, yeah. Yeah.
We can't even layer that into spatial transcriptomics because we've tried, right? And it helps, but it doesn't because it's little dots. And they don't know what to do with teeny tiny dots because they're used to the whole cell lighting up a color, right? And so, everyone saw those beautiful images of xenium or cosmics. Like, we think that's amazing. But if the person who has to interpret them they don't even want to see it. They only want to see 3 colors. And even then they want it separated out to brown dots. So it's not a trivial task to say, I mean, it's amazing. We have whole transcriptomic level now. Imagine how many cell types we're going to have. They don't even want to see past 7 antibody stains, right? No one see past 3. So conceptually, it's not a small thing to say, how do you transform transcriptomic level spatial data into the pseudo dots that we're talking about proteomics, right? It's not a trivial task.
I mean, even in that aspect, we may have to educate the pathologist as well. It is not the total number of T cells in a tumor that determines the response to treatment. It is where exactly they are located. If they are infiltrated within the tumor, the response is better rather than they are just sitting outside the periphery. But in our single cell studies, you are going to still find a lot of T cells in the tumor, but it doesn't respond, right? So we need to integrate the spatial with the whole single cell and then the location. These are the 3 different things. I don't know how we can make a pathologist or somebody integrate.
Well, I would say it's not just the pathologist problem, it's our problems.
Oh, yeah. It's
We
our
actually lead the problem because what I encounter the most being a single cell person, having adopted spatial a long time ago, it's the wrong mentality.
Yeah.
Think about, you have to be thinking about the spatial data first.
Yeah.
If you're trying to be a single cell person and annotate, like you said, it doesn't matter.
Yeah.
We're counting T cells proportionally and saying that's why immunotherapy is not working. But you physically look at the patient and there's a blockade of fibroblasts.
Yeah, exactly.
No matter how many T cells you counted, right?
Yeah.
But that's the onus on us before we even think about the pathologist. And we're not there yet because most of the software tools now are, how do we take all this amazing single cell data and make it annotated into our spatial? And that's the absolute wrong approach.
I mean, essentially, it looks like we need 3 or 4 different ways to build this atlas. One, we need the single cell or single nucleus, then the spatial, then the pathology, and maybe there may be something else. So I don't think the things are simple, but we need to start somewhere, right? So let us move on to the number 5. Again, this is an area we still are working on, the inferred ancestry, which keeps changing all the time. There was a paper which came out I was showing somebody yesterday that within the African ancestry, East versus West African ancestry, show a completely different relationship to certain disease parameters. So how do we, to what granularity we need to include the ancestry in our cancer map? Any thoughts? Anybody have any thoughts on that?
So I think we report on a continuous variable. We report it as a continuum rather than as categorical, whatever the ancestry is. And that's why we need lots of numbers so that we can tease out that nuance, East versus West, North versus South, and even within like Asia, right? We saw that this morning. That it's how many billions of people and very diverse, but not as diverse as Africa, for example. So it's a lot of data, and it has to be a continuum, and not like we want it to be easy by just putting categories of Black versus white, but that's not going to give us the answer, especially as we become more and more admixed.
In this sense, for instance, nowadays HCA data is divided in 18 bionetworks, mainly by organs, right? How would you guys envision the cancer atlas divided? Like, how would be this organization? Would be by organ? Would be by disease subtype? How do you see this? And also, on top of that, if we need to prioritize a cancer type, how we would do this choice?
I have the mics. I'm going to talk.
Please, do it.
And that's why I brought up this slide, which I didn't show before. And it is not going to be easy. The situation, I mean, however, it is not simple, because this is the paper which was mentioned this morning. We had it here, even in Brazil, one country, and look at the admixture population. And according to that science paper, this admixture has several genes associated with fertility rate, immune response, and distinctive metabolic traits. So within one country. So how do we incorporate that into our cancer atlas is something we really need to think about it. Hard, all right?
So it's capturing the metadata also, like what we presented this morning, the tier 2 metadata. It's a nice to have, but we should have it. The exposures, for diseases that affect women, we should have fertility, reproductive factors, because we know those are drivers in at least from the perspective of gynecologic and breast cancers, and maybe some other cancers too. They're hormonally driven. But as you're asking about how do we organize, it's by one organ and also disease type. It depends on the question. But once you get the tissue and you have the metadata that you get also from the pathology report and all those things, then we can start like reclassifying like neuroendocrine tumors, independent of tissue of origin. There are neuroendocrine tumors, like there's doctors that just treat neuroendocrine tumors no matter where they come from. But you can only get to that point if we start like capturing that data. So it's side disease groups, which is, if we want all of this to be translated into like humans and treating diseases or preventing diseases, we have to kind of mirror what's happening in it.
More thoughts on that?
Yeah.
Thank you. Regarding the type of tumor, I think that probably something that has similar frequencies across countries, but with different outcomes regarding fatality or regarding response. Because, for example, in Mexico, I am from Mexico, so we know very well that our rates of success for patients with leukemia is very different. It's inferior to responses in other countries that have the same type of leukemia. So probably picking one tumor type that is occurring across all of the countries that you might have access to, and with different outcomes. That will be my bet, I guess.
I think you brought an important point here. I mean, I think since we cannot cover every cancer and every, so first we need to talk to the epidemiologist to see which cancer in which country has a different outcome. Then maybe the investigators in that particular country focus on that, and using all these technologies we see, I mean, we presented here, and then come up with a map to solve that rather than making a global one cancer, everybody work on one cancer. Maybe it is the country specific.
Yeah, because in this specific case of leukemia, I am thinking that it's not that, well, the one hypothesis is that it's not that we're really genetically very different and then we're not responding, but it might be the probably the delay in the diagnosis of the patient, for example, or Mexico is a very, it's a country with big contrast.
Yeah.
So very rich people, very poor people.
Okay.
Even though we might be all of us mestizos, there's rich people, poor people, then the poor, they will have less access.
Yeah.
So I'm just thinking of leukemia, but can be something solid tumor for applying spatial transcriptomics.
Right, right.
Thank you.
I mean, there are certain countries with the GI tumors being much more predominant than other countries, right? So people always think it is their diet or whatever it is. So in focusing country, the cancer by country may be a better option, I believe. Anybody else have any thoughts? So the last question here, on this page, should we use whole transcriptome spatial profiling or disease-specific targeted panels? So the question is, the panel can be designed based on the single cell, single nuclear data of the tumors, or should we go all over again starting?
In this case, let's think that money is a constraint.
Yeah.
So if we don't have all the money that we want, can we think about a minimal panel that could be used to compare results among different projects, different tumors?
Or skip the whole step and get into the protein, as it was said before.
Okay.
So the question is which way we should move forward.
Yeah. What do you think?
Yeah.
For instance, please.
Mine's just a comment. I think the best predictor for therapeutic outcome to most diseases is your postcode, right? It's where you live, right, in the world. Most countries will not have access to immunotherapies. If you take Southeast Asia, that you are using a drug for cancer that was given 20 years ago, it's off patent, and now it can be given. So I think the question needs to come back to a fundamental biological annotation of the disease, because that is universal, right? That we can control. Today, patients are given ADCs, they're given IO, but in most countries, you won't have access to those drugs. So it's just trying to frame, are we thinking of in the context of IO, or maybe dialing it back and trying to understand the disease? For the 0.6, I'd say whole transcriptome, you'd have to get that level of annotation integrated with your H&E. Without those two layers plus protein, we do same slide, whole transcriptome, H&E as a base, and then you add in your protein. And if you can do all of that on the same slide, that's even better. You have all those data sets.
Just to clarify, when you guys are talking about protein, you are thinking also about immunohistochemistry, like just one antibody, one marker, one protein, or are you talking about panels of proteins, like more expensive experiments?
And also 30 to 50 markers, single cell. Yep.
I think hopefully if that becomes maybe another 10, 15 years, it becomes affordable like H&D, where you can do the multiplex for say 30 to 40 proteins, and you don't need a sophisticated $1 million microscope to study that. Probably that's when I think all this become practical. There are a couple of few other thoughts we put in together again. I mean, the roadmap which came about by the from the immune cell diversity. Something we learned from that, can it be applied to the cancer field? I don't know anybody who was in the immune cell diversity classes here in the room, how difficult it was. And the second question we have is again the adaptive traits that I mentioned. To what extent we need to give importance to that while defining the cancer atlas? One for sure, the one which I emphasized on the Duffy trait, we all know that the women who are null for the Duffy, although the gene is called ACKR1, if they have the polymorphism, they don't express that gene. So then they are protected against malaria, but if they get breast cancer, their breast cancer tend to be much worse. So to what extent many of the adaptive traits that is in the literature should we take into consideration in the cancer field? That is something, a question we need to be addressing sometime.
Any thoughts on that?
I mean, these are some of the questions. We'll make it available for you guys. And if you want to adopt it in your own research, you can take that. And again, to what extent these traits has to be taken into consideration while interpreting results of single cell and spatial transcriptome studies. If you have some outlier data in your spatial work or the single cell, do you want to link it back to your adaptive traits? That is something we may have to consider as we move forward. And again, the immunotherapy came into picture. Few of you have already talked about it. Again, the response varies as well as the toxicity, cytokine storm. When we do the cancer atlas, what other parameters we need to consider, which can tell us about the potential toxicity issue with the treatment. So these are some of the questions we both thought about it, and you may have other questions. I think, I don't know how much time we are left with, maybe another 5-10 minutes, maybe.
15?
Another 15 minutes. So now again, any of the things which we didn't put it up here, please bring it up.
Okay, there is one comment from
Okay.
people online. I'm gonna read. For cancer patients, how the inter and intratumor heterogeneity can be addressed with current technologies. Most computational methods harmonize the data. How reliable are those? Does someone want to comment?
One of the things people have been using is the inferred CNV from the single cell data. Single cell nuclear or single cell transcriptomic data, where you can use programs like Copycat, where you can infer the CNV. So that will allow you to figure out the heterogeneity within the tumor, right? Pre and post treatment. I don't think the spatial will help in that direction, but the
Can apply the same methodology to spatial. To infer copy number variation.
Exactly. If you go the transcriptome route, you will be able to.
Yeah.
Yes, exactly.
Yeah, I'm thinking about spatial transcriptomics.
Yeah. Again, everything is inferred. Copy. Yeah.
Please.
Hi, thanks. Great discussion. The question depends on why you want to build an atlas and what you want to regress out when you build an atlas. Is the patient heterogeneity something that you want to regress out when you build an atlas, when you pull so many data sets into one joint space? And it also further extends to what kind of technologies do you have? Like if you have single cell, you don't retrieve cells like neutrophils, but you have single nuke, you don't retrieve immune cells. So I think the ideal way to go about this would be to integrate data sets based off of using the donor as a batch. So you have some anchor and some common point that you can use as a way of reference, and then to impute spatial on top of it. So it's also cost effective and also has much more added value. Thanks.
Thanks. Anybody else have any comments?
I think spatial is expensive today, but you have to get creative in how you build efficiencies with it. One way we do it is we build tissue arrays, 100 cores per slide, and now your $5,000 experiment, you have 100 patients on a single slide. We do 2 intratumor, 2 peritumor, you get 4 cores per block. There are ways in which you can sample. We get a pathologist to go and annotate multiple areas
Are
within the...
you talking about tissue microarray?
Yeah, but you get the pathologist to build that where they select 2 intratumor, 2 peritumor, and now you've got 4 cores per patient. You put 4 onto that, you fill that slide with another 96 samples, and now you've got 100 samples, single TMA. That's how we've done our 600 patient sample cohort. Reviewers and people will tell you, yes, you're looking at a TMA versus not a whole slide, but you're not going to do 600 patients at whole slide today. So there are trade-offs, but I think you can get a lot more tissue on using arrays. All our tissues come from Brazil. I mean, they're beautiful TMAs. They make them all for us from Curitiba. We did the same for COVID, lungs, hearts, brains, placentas, all from, and even with TMAs, you can go from 0.6 to 2 millimeter cores. So you can get spatial data from that. So
Perfect. And remind me again, how many areas do you select in general?
2 intratumor, 2 peri.
Okay.
So 4 per block. Yeah.
Perfect.
There is a question over there, comment over here. Yeah. Jasmine.
I mean, I am on Arutha's train. We routinely look at 20 million cells, right, on one tiny little study. So the idea that, a TMA is not going to give you anything, we get over 10 on the tiniest 0.2 millimeter punch. We're getting over 10,000 cells. I would argue to anybody in the single cell atlas that they're getting more than that off the old data that we were used to generate. So you even expand that out to a 1 millimeter punch, we're getting 50,000 cells, right? Now we can actually replicate within the tissue. So you're saying we're not getting the whole baked tissue section, but we're going to be able to take, 2 or 3 punches from that one patient. So on a given slide, we're profiling 2 million cells, 3 million cells. So that's how you get to 600. So I would argue kind of the idea that the maps, Tabula Sapiens got published at 1.2 million cells only 3 years ago. I do that on one slide on any given day.
I agree with you, but if there is a pathologist in a study section who is of a old school of thought, your grant will get triaged when you do that. You did everything with the TMA. That's based on my own personal experience. So that's a different story, okay?
I would say there's enough space. We're talking spatial now, but there's enough spatial data. And I think Arutha and I can talk about it, but anyone else that has publications out, I would say a few years ago, but we are getting routinely, maybe there's the old school group that, of course, we are getting a lot of have to go through reviewer 5s now, right, in spatial data. But that's because I think people are realizing you can't come after us for numbers anymore. That's a very old idea because you were based on H&E, and we have gene expression now and whole transcriptome gene expression. So I think that we have to be very forward thinking, especially if you're trying to build a comprehensive map. I think that that's no longer true. I mean, we're examples that you can publish off that, and we are getting more money, more bang for our buck, and more cohorts done this way than, what was done for the Goliath task we had for the first map or atlas.
Here is one more comment. Please go ahead.
Yeah, like adding up a little bit on the whole slide versus TMA, I think that obviously when you go for whole slide, there's a lot more information, a lot more things you can get out of that, but obviously it's not feasible when you try to make it more high throughput.
Yeah.
So I think there should be a lot of attention put on statistical methods that help you guide more how you select those TMAs. And that's particularly something that we're talking about space and the way that we are analyzing space. There's many sampling methods that have been used throughout history. For example, in ecology, when you want to sample a region, that can be leveraged a lot in how we address the way we are analyzing these samples. And I think that's what the spatial dimension is about, like taking that into account and actually having representative sampling when we do TMA, because otherwise doing whole slide right now, it's not for many laboratories feasible as it should.
So if I understand correctly, these in the spatial world, the statistical methods have been pretty robust, those who have been using it, right? But
They are, but I think he's mentioning about representativeness of the tumor sample, right?
yeah.
Yeah.
So
So in this sense, do we agree as a community that ideally we would work with the entire sample, but in cases of money constraints, then three points of areas that really represent the tumor would be a good way to start profiling those samples?
Yeah, I just have a comment with whole slides. So whole slides, you're assuming you're going to get 3 million cells. In that biopsy, if you get 500,000 on slide 1, 5 million on slide 2, slide 3 and 4 are arbitrary numbers. Now, how are you normalizing? How are you batch correcting? Also, you're running 4 slides at a time. You might come back the next day and run the next lot. So there is so much incredible variation in antibodies and transcriptome data. If you look at and so this is why TMAs are great, because it's 100 patients, one slide. Tonsil on the back end, we cut tonsil at the bottom, so we have some sort of control within that. So, ideally, the way we're thinking about scaling spatial by whole slides because of technical issues doesn't work, because of batch effects, and there's no way that no one has shown any way to normalize across 100 whole slides. There's now, and because you've got confounding data with cell numbers, and if you've got a tiny biopsy in a huge resection, how are you comparing? There's no way. 5 million cells versus 100,000, you can't do it. That's why 50,000 cells per TMA, reducing your number of slides, getting more tissue onto fewer slides is probably the easiest way forward initially, until we figure out how to do the bigger whole slide.
Perfect. Thank you.
Another 5 minutes we have, right?
Well, maybe one subject that we didn't touch is about the 3D spatial configuration. Do you guys can see the benefit of using this kind of technology to understand spatial cellular interactions, for instance?
I mean, we have heard a fantastic talk about the autism, where they're making so much inference out of the iPS cells. Do we think we have something equivalent to that to go ahead with the tumor?
How do you think this could help us to
Make
move
it
forward?
practical. Yeah, make it more practical.
Well, yeah, and lots of AI to reconstruct. Yes.
Yeah. Again, any thoughts you guys have, if you want to send us an email, just let us know, right? I think it's a productive conversation, but I think it's the very beginning. We have a long way to go with this. But we all acknowledge that what HCA has done with the normal should always serve as a reference for the cancer atlas we want to build in future. So HCA's contribution is unparalleled in this regards in setting up the normal, right?
Yeah, please.
Yeah.
Yeah, I was just wondering if anyone had any thoughts about generative AI, which is able to generate sort of like H&E slides from, say, other H&E slides or from spatial transcriptomics data. Would that help in various ways? For instance, I've seen some groups using Gen AI generated H&E slides to then further train an AI model. Or could you also use this kind of Gen AI methods for various other applications in, say, helping pathologists to diagnose tumors, etc.?
Yeah, I mean, there's a lot of push in that direction. Particularly, I have a collaborative grant with from DOD, where the pathologists are now using H&E stain slide to figure out the collagen orientation within the tumor. I know the Google has talked about the AI pathology for the last 10 years. They spent so much money on that. There are a lot of publications came out, but I think there is it's still not being used in a routine setting in the clinic yet, right? So it will be there for sure, but I don't know how long it's going to take.
Any topic that maybe we didn't cover, but
Yeah,
you guys
exactly.
would like to talk about? Yes, please.
Please.
Yeah, so I just thought if making a comparison to a couple of years ago before the single RNA sequencing data became available, I mean, the Human Cell Atlas with all these meta-analyses, what we've been doing for the past couple of years, something very similar will likely happen with the spatial transcriptomics, that the consortium will not really have much control over what people in the field will generate, and there will be this large body of work. And the question is, what can be integrated, and sort of the panel designs, but also the questions around the tissue arrays versus the pool. And so there's a range of very technical questions there about how to construct an atlas from the data that's available and assessing what makes sense to kind of pool together.
So I mean, yeah, I mean, you brought an interesting point, whether can we get it done without even doing a single new experiment? You know, there are people who are already doing the single cell work with the cancer, different cancers. They are out published. Here, HCA has created the normal one. Should we try to integrate two and come up with a new thing on the cancer? Do you think that's feasible?
I don't know, but I think we should find out.
Yeah. I mean, there has to be some collaborative efforts between the HCA and those who have created a lot of cancer atlas so far, right? That may be something to think about, the meta-analysis of both. Right?
Yeah, I think what we need to think about, and I think this is our may goal when we return to our homes, is to think how we want to see this atlas, this cancer atlas, how this is going to be a representation of us, right?
Yeah. And of course, we should not lose our focus on making sure that it involves the diverse population. That's why it is diversity, disease, cancer, this particular, right? So.
Yeah, okay. So
with that, thank you all for participating. Really appreciate it. Hopefully.
thank you for the discussions. Thank
Yes.
you all.
Yeah. Okay, there was a chat. This we already gone through, right? Yeah.